Continuing with biomolecules, our next topic is alpha amino acids. These are the organic compounds having both amino group as well as carboxylic group. Since amino group is present on the alpha carbon, that's why they are called as alpha amino acids. This is alpha amino acids. Each alpha amino acid is having a chiral carbon, due to which it is optically active. Its optical activity can be shown like this: HNS2, R group, and COOH. They are kept on the vertical side. H rotates light from H to NH2 group. Similarly, in this case, light is rotated in a anti-clockwise direction, so this is minus alpha amino acid. This is plus alpha amino acid. So coming to the class of alpha amino acid, all alpha amino acids belong to L series, because NH2 group on penultimate carbon lies on left hand side, just like L-glyceraldehyde. This is CHO. Then we have amino acids. They belong to L-alpha. L-glyceraldehyde is a derivative of methyl group obtained by substituting one hydrogen with vice group. This is L-glyceraldehyde. Now there are total 26 alpha amino acids known till date. Out of which 16 alpha amino acids they can be synthesized by our body. That's why those 16 alpha amino acids they are called as non-essential alpha amino acids. While 10 alpha amino acids they cannot be synthesized by the body. We are totally dependent upon the external sources. Therefore, they are called as essential sources, essential alpha amino acids, since they are to be taken from outside. So, alpha amino acids they are of 26 types, and their nomenclature will be discussed later on. First three letters of their name they are introduced in their representation, in their abbreviation. They become the representation of alpha amino acids. Then we have structure of alpha amino acids. The alpha amino acids they behave as amphoteric compounds. They can accept proton. This is intra-condensation, you can say, or intra-migration of hydrogen from carboxylic group to NH2 group. Since NH2 group is having a lone pair of electrons, therefore it can accommodate the polar H positive ion from carboxylic group and can form NH3 positive. So alpha amino acid they exist as filter ion. This is intra-condensation or intra-migration. This hydrogen migrates to nitrogen, you will be getting NH3 positive. This is called as filter ion or dipolar ion. All alpha amino acids they exist in this form. It is due to internal migration of proton from carboxylic group to NH2 group. Then we have very important property of alpha amino acids, that is your isoelectric point. All alpha amino acids at a particular pH, either cationic part or it is anionic part which dominates, and these alpha amino acids, they start migrating towards cathode or anode under a particular pH. The migration of this alpha amino acids towards cathode or anode under a particular pH, that will take place, that is due to the unequal number of cationic part and anionic part. But sometime when sometime the amino acids, they do not show any migration towards cathode or anode, and that pH of the protein solution or alpha amino solution is also called as the isoelectric point. This is the pH alpha amino acid solution or protein solution under which does not show migration towards cathode or anode. This pH is called as isoelectric point of your alpha amino acid or protein. Different alpha amino acid, they have different isoelectric point. Example is glycine has isoelectric point of 6.1. Then we have aspartic acid, it has 3, lysine 9.7. Aspartic acid has isoelectric point of 3. Then we have lysine, it has 9.1. So different alpha amino acids, they possess different pH under which they do not migrate towards the cathode or anode. This is mainly because of their equal number of cationic or anionic part in the alpha amino acid. Then we have the nomenclature of alpha amino acid. The names of the alpha amino acid, they are represented by first 3 letters of the name of the alpha amino acid. For example, we have glycine, so its abbreviation will be GLY. We have lysine, iso abbreviation will be LYS. We have alanine, abbreviation will be ALA. We have glutamic acid, abbreviation will be GLU. So first 3 letters, they are generally taken. If third letter is repeated, then we take the fourth letter. Similarly, we have leucine, abbreviation is LU, isoleucine, ILU. And then we have proline, abbreviation is PRO, phenylalanine, PHE. And then we have methionine, MET, tryptophan, TRP, serine, SCR, then cysteine, CYS, aspartame, ASN. Then we have threonine, THRE. Then we have tyrosinase, that has T by R, glutamine, GLU, GLN, then cysteine. Then we have indogorgic acid, hydroxyproline, threonine, aspartic acid, glutamic acid, arginine, lysine, histidine. So they have the abbreviation of first 3 alphabets of their names. Then we have, after alpha amino acid, we come to the topic what are peptides. Peptides, these are the condensation products of alpha amino acid formed by condensation between carboxylic group of one alpha amino acid with NH2 group of another alpha amino acid. This combination is accompanied through elimination of water molecule, and a peptide containing 2 alpha amino acid is called as dipeptide. If it contains 3 alpha amino acid, it is called a tripeptide. If it contains more than that, similarly it will be classified. Nanopeptide, for example, has 9 alpha amino acid, octapeptide has 8 alpha amino acid. So in this manner the peptides are being classified. How they are formed, let us see their formation. This is our first alpha amino acid, combines with hydrogen of NH2 group of the next alpha amino acid. This eliminates the water, and n number of units they combine, we get n number of peptide linkages. This is called as peptide bond. This is our peptide bond. This is dipeptide, since it is made up of 2 alpha amino acids. When number increases, their molecular weight also increases. So a peptide with molecular weight more than less than 10,000, then it is called as peptide, simply polypeptide. If molecular weight of the peptides increases, becomes more than 10,000, then these polypeptides, they are started calling as your proteins. So proteins are the proteinaceous substances. They are the big macromolecules formed by combination of large number of alpha amino acids, and when the molecular weight of these polypeptides that exceeds 10,000 EMU, they are termed as proteins. So biologically important proteins, let us see some important polypeptides. First of all, we have oxytocin. It is a peptide hormone. Produced by posterior lobe of the kidney, pituitary gland, I think. The function of oxytocin is lactation of mammary glands during childbirth, production of milk, and it is also responsible for contraction of uterus after childbirth. Then we have second polypeptide, that is vasopressin. It is also a nanopeptide formed by combination of 9 alpha amino acids, also produced by posterior lobe of the your pituitary gland. It is responsible for the reabsorption and control of the water absorption by the kidney. Then we have third important polypeptide, that is angiotensin 2. It is a octapeptide. It is found in blood plasma of those persons who are suffering hypertension, who are the patient of high blood pressure. It is found in the in blood plasma of those persons. So this is regarding the alpha amino acids, peptides, and important peptide hormones we have studied. They are actually oxytocin, vasopressin, and then angiotensin 2. After doing this, we come to another important topic, that is proteins.